we've heard a lot about immunosenescence and how we may or may not want to define it. I'm going to be using a rather narrow definition in this talk. But we've heard how immunosenescence could be a problem as we continue to increase these T cells that are essentially useless and in theory take up this so-called immunological space. So what we did is we decided to try to actually do something about this, and that's what we did. So I'm not going to really go through this. This has been touched on in detail by earlier speakers. As we know, vaccine response really declines in the elderly. Between 30 and 75% of older people fail to adequately respond to flu shots, and according to some more recent papers I've read, that maybe even higher. Allergic T cells, as we get older, they expand in number. Certain CD8 populations, as the last speaker just went over with in detail, the CD8 cytotoxic T cells, they tend to lose their CD28, which is a co-activator, and they become not nearly as useful as they formerly were. In mice, the marker KLRG1 is highly associated in the same manner. Also in humans, but perhaps to a lesser extent. And we've gone over immunological space to some degree with earlier speakers. So, KLRG1 in mice. Now, this is from the literature. We see that young mice, they have essentially no KLRG1 expressed on their cytotoxic T cells, and as they get older, it increases. There's as you can see just from this, there's a tremendous amount of variability in that, as we would expect. And when we tested this ourselves, we indeed found the same thing. Mice that are just 3 weeks old, they have almost no KLRG1 expression on their CD8 cells. When they're about a year old, it goes up to over 10% on average. So the first thing we tried to do something about this was we directly injected KLRG1, an antibody to KLRG1, into these mice, in effect to try to generate an immune response against these cells. And we did have a response, but we more or less abandoned this approach because of concerns of cross-reactivity with the antibody and other cells. For example, KLRG1 is found on our native NK cells, normal ones, and alternately spliced versions are found on other cell types like mast cells, etc. So we abandoned this approach because it seemed like too much of a shotgun. So we instead tried to look to some of these analytic techniques that they use for cell separation, only use them in a therapeutic manner. Flow cytometry is commonly used for analysis. We actually decided to go with magnetic cell separation. And the way magnetic cell separation works is you selectively tag cells with superparamagnetic particles bound to antibodies, and then separate them by applying a magnetic field and then washing away the untagged cells, and you can obtain extraordinarily high purities of whatever cells your antibody is against, whatever marker your antibody is against in the cells that express that marker. In our case, we used the biotin stripped out and bond to bind the magnetic particles to the antibodies. Applying the same idea at a much larger scale gives therapeutic applications. For example, this has been used in humans to remove cancer cells in autologous bone marrow transplant when other adequate bone marrow is not available. So without going into any more detail there, our procedure in a nutshell is this. We removed blood from mice, mixed with some of these KLRG1 antibody, some of this KLRG1 antibody conjugated to these magnetic particles. And then using a big rare earth magnet, we pulled these labeled cells to the bottom of a little tube and pipetted out the blood and discarded the pellet with all these KLRG1 positive cells. And we then sent this for analysis to see what was actually remaining in our blood after this procedure. Before I go any further, I just want to give you a little short video clip of what magnetic separation looks like. This is just the magnetic particle suspended in solution and holding a magnet against it. And as you can see, it goes from being, muddy water to being a concentrated magnetic pellet. And the exact same thing happens when we do this with blood. So this was our result in isolated blood. This was just blood. This was not in whole animals. And on average, with these samples, it was a little over 12% KLRG1 positive CD8 cells, and after the procedure, we had less than 1% positivity for KLRG1 on these CD8 cells. And then the next step was to try it in one whole animal. So that's what we did. We tried it in one whole animal. And we did this by removing about 20% of its blood at a time. This blood was treated the same manner I just described earlier, and then reintroduced to the animal, and then the animal was given 15-20 minutes, and then this process was repeated 15 times. And then several hours after this treatment, we took our blood sample, and this is the result from this one animal. And that animal started with about 6.5% KLRG1 positive CD8 cells, and after less than 1%, we had about a 7-fold decrease. So this was encouraging, so we decided to go into the next step. And that was a device that would do all this really boring work for me. So this is just one of our very initial designs. It probably has not much to do with what we actually did. But yeah, we built a device. And to help us do that, we brought in a roboticist, Keith Causey. He was actually a great worker. He didn't know much about biology to start with, but we taught him the few bits that he needed to know and how to work with mice. And after just a few weeks of this, he essentially took over the project. And all the data you see after this was thanks to Keith. So let me show you a little video we have of our device in action. Let's see here. So this is a mouse. It has restrained. It has a jugular venous catheter, and the device automatically withdraws a predetermined amount of blood from the mouse. It draws it into this mixing chamber here where the nanoparticles are located with the antibody. There's the magnet, pulls away. It mixes for actually much longer than is shown here. We then remove it, and after being treated 15 times, this is the mouse about 2 minutes after having this treatment done. It's completely fine. So this was what this mouse looked like before, with its CD8 cells monitored on the x-axis and KLRG1 on the y. The cells that we're interested in are right here in this little R6 box. And you can see that after treatment, the R6 box is essentially empty. And the cells we were after, we managed to take away. So we did this in a bunch of animals. And this is the data we obtained from that. On average, these mice from this experiment had about 17.5% KLRG1 positivity in their CD8 cells. And afterwards, it was up to about 2% or so, or about a 7-fold decrease again, which